It is widely accepted that our species, or at least our lineage, may be approaching 1 million years old and originated in Southwest Asia, or more precisely, the Levant region of the Eastern Mediterranean. The words of one British paleoanthropologist are very revealing. It is difficult to discern a clear pattern of change through time from African fossils to early Homo sapiens fossils outside of Africa. This admission is striking because it reveals that the search for the earliest Homo sapiens in Africa shows no continuity between the famous East Africa fossils such as Hairto and the first Homo sapiens of the Levant such as School and Qesem. However, when we look closer, we do see continuity, but not where it is supposed to be. We see an early Mediterranean Homo sapiens population with the teeth, jaws and marine-based diet that would lead to anatomically modern humans. Teeth do not lie. Teeth do not care about ideology. They do not bend to migration models, nor to the simplistic arrows drawn on maps. Enamel mineralizes early, resists decay, and outlasts bone. It preserves cusps, grooves, roots, and wear long after cranial vaults collapse and narratives shift. If we want to understand deep population history in the Mediterranean corridor between 300,000 and 90,000 years ago, we should begin with the teeth. And when we do, something remarkable emerges. A thread of morphological continuity stretching from Qesem to Mislia, Tabun to School and Qesem. The pattern is not one of isolated failed dispersals or clean species replacements. It is the signature of a long-lived Mediterranean metapopulation. Start at Qesem Cave, dated roughly between 420,000 and 200,000 years ago. The hominins there present a mosaic. These teeth show reduced molar size compared to earlier Middle Pleistocene hominins, relatively gracile crowns, and patterns of enamel thickness trending toward later Homo sapiens. Qesem is not an outlier. It sits geographically and morphologically between Africa and Eurasia, and its dental pattern anticipates what appears later at School and Qesem. There is an important biological reason why this matters. Faces and brain cases are relatively plastic structures. Cranial vault shape can respond to developmental shifts, climate, even subtle changes in growth timing. Facial projection, brow ridge thickness, and cranial curvature can vary under environmental and hormonal influences across surprisingly short evolutionary spans. Teeth are different. Once enamel forms in childhood, its morphology is locked in. Crown shape, cusp pattern, root configuration, and enamel thickness are strongly genetically controlled and far less responsive to environmental pressure. Diet can affect wear, but it does not redesign cusp architecture. Climate can influence body proportions, but it does not rapidly reshape molar roots. Because dental morphology evolves more conservatively and resists environmental remodeling, it is one of the most reliable indicators of lineage continuity in the fossil record. When teeth align across time and space, we are almost certainly looking at shared ancestry rather than superficial adaptation. A metapopulation is not a single homogeneous group. It is a network of subpopulations exchanging genes intermittently. In such systems, traits can persist regionally while being diluted elsewhere. Reduction trends can spread gradually. There is no sharp boundary where one lineage ends and another begins. If Homo sapiens sensu lato, meaning the broader lineage that gave rise to us, extends back toward 800,000, then the search for origins shifts from a narrow East African window to a broader North Africa Near East zone. The Levant and North Africa become part of the same evolutionary theatre. Teeth don't lie because they cannot easily converge under superficial adaptation. Crown shape and root structure are developmentally constrained. They reflect ancestry more than environment. When we see repeated patterns across time and space, we are seeing lineage. This brings us to something else. Fish. In the Levant, researchers analyzed fish teeth recovered from archaeological layers associated with controlled fire. The enamel microstructure revealed exposure to temperatures between roughly 200 degrees Celsius and 500 degrees Celsius, far below the temperatures of direct flame, but consistent with slow, controlled heating. The teeth were not calcined or cracked in the way bones tossed into open fires typically are. Nor were they charred black. Instead, they showed the thermal signature of indirect heat. suggestive of cooking in embers or, more intriguingly, in earthen ovens. The pattern is subtle but decisive. If people were merely throwing fish heads into the fire after eating raw flesh, the enamel would show higher temperature shock and direct combustion signatures. Instead, the fish appear to have been heated evenly, likely wrapped or covered, perhaps under leaves and sediment, in a pit oven. This implies not accidental burning, but controlled culinary processing, and it implies planning. The two species identified, the Jordan barbel, Luciobarbus longiceps, and the Jordan himri, Charosobarbus canis, are not random fish. Both belong to the Cyprinidae family, freshwater carp-like fish that inhabit rivers and lakes in the Levant. Today they are found in slow-moving freshwater systems, including river channels, lake margins, and vegetated shallows. Importantly, these fish are relatively large and nutrient-rich. The Jordan barbel can reach lengths exceeding 50 centimeters, nearly 2 feet. A single specimen provides substantial caloric return, especially when cooked whole. Cooking softens bones and connective tissue, allowing marrow-like extraction of nutrients and making even small bones digestible. That could explain why fish teeth survive archaeologically, while many bones do not. Low-heat cooking may have rendered bones fragile and prone to disintegration. The absence of extensive fish bone accumulations at the site may actually support the earthen oven hypothesis. Slow cooking at moderate temperatures, perhaps wrapped in leaves or buried in embers, would preserve enamel but degrade delicate skeletal elements. Teeth with their highly mineralized enamel are far more heat-resistant. Once again, teeth do not lie. They preserve ancestry in enamel and dentin, and when we follow that trail across the Mediterranean, the picture that emerges is not one of isolated species flickering in and out of caves. It is a long, complex, resilient metapopulation shaping the deep history of Homo sapiens. What makes this discovery explosive is not only its age, 780,000 years ago, but its implications for who these people were. The eastern Mediterranean at that time was not a peripheral zone. It was the hinge between Africa and Eurasia, between Mediterranean coastlines and inland savannas. It was also ecologically rich: freshwater lakes, wetlands, fish, birds, and plant resources. If early members of our lineage were cooking fish here nearly 800,000 years ago, we are looking at populations with controlled fire, food processing knowledge, and dietary flexibility long before the traditional behavioral revolution narrative places such capacities. The Mediterranean was not a barrier but a connective ecosystem. Coastal routes, shared faunal communities, and similar climates linked Morocco and the Levant. Rather than imagining isolated African origins followed by a single late exodus, it becomes plausible to envision a Mediterranean metapopulation, groups distributed along coasts and river systems, exchanging genes and ideas over hundreds of thousands of years. In that framework, the Eastern Mediterranean and Moroccan hominins are not separate evolutionary experiments. They are branches of the same deeply rooted population network. The key to understanding what this means lies in diet. A consistent seafood diet changes biology. Fishing requires unique skills and less brute strength than hunting large, dangerous animals. This also leads to more gracile bodies, less reliant on testosterone and adrenaline, and leans more towards patience and ingenuity. In fact, the first projectile spears may have been invented for fishing rather than terrestrial hunting. One of the major biological differences between Homo sapiens and Neanderthals is the substitution of the amino acid lysine for arginine in the brain. It has long been argued that a marine-based diet could have led to a sudden spark in intelligence that separated Homo sapiens from other humans, so this is not pseudoscience. The problem has always been a lack of archaeology and fossils in Africa to prove this theory. Marine and freshwater fish provide long-chain omega-3 fatty acids, such as DHA, essential for neural development. They also supply iodine, selenium, zinc, and high-quality protein. Importantly, fish are rich in arginine, an amino acid central to nitric oxide production and vascular regulation. Arginine plays a role in cerebral blood flow and synaptic plasticity. A diet increasingly dependent on aquatic resources may have reshaped metabolic priorities in early humans, favoring enhanced neural vascularization and sustained brain energy supply. Cooking amplifies these effects. Thermal processing breaks down collagen, softens tissues, reduces parasites, and increases caloric accessibility. It decreases digestive strain and reduces the need for large jaws and massive teeth designed for raw mastication. Over evolutionary time, populations that regularly consumed cooked, soft, nutrient-dense foods would experience relaxed selection on heavy chewing apparatus and increased allocation of energy toward neural tissue. The archaeological record shows precisely such trends. Across the Middle Pleistocene, hominin dentition becomes smaller, jaws become less robust, faces begin to retract, cranial vaults expand and round. These changes are not instantaneous. They are gradual, cumulative, and correlated with shifts in subsistence strategies. A seafood-based cooked diet along Mediterranean shores could help explain this transition. Rather than seeing gracility as a sudden late Pleistocene innovation, it may reflect a much deeper dietary pivot, one anchored in coastal ecologies nearly 800,000 years ago. The evidence demonstrates controlled heating at moderate temperatures, not wildfire opportunism, but deliberate culinary behavior. Earthen ovens represent a technological step beyond simple fire use. They require knowledge of insulation, timing, and fuel management. They imply social coordination. They suggest repeated practice. And importantly, they suggest foresight. This is not a population barely surviving on scavenged marrow. This is a group managing energy resources, exploiting aquatic ecosystems, and optimizing nutrient intake. This is not a failed migration from a mythical African homeland. If the Thomas Quarry hominins in Morocco represent a population near the root of later Homo lineages, and if the humans 780,000 years ago were cooking fish, then we may be seeing the metabolic foundations of Homo sapiens long before our textbook origin dates. Consider also the dental implications. Human teeth do not evolve rapidly under environmental fluctuation unless selective pressures change diet. Softer cooked food reduces enamel wear and decreases the advantage of thick enamel and heavy occlusion. Over hundreds of thousands of years, dental reduction becomes stable. This is precisely what we see moving from earlier Homo forms toward later Middle Pleistocene populations. Similarly, jaw reduction follows. A decrease in masticatory stress reduces the need for large mandibular bodies and muscle attachment sites. Faces become more gracile. Brow ridges diminish as cranial architecture reorganizes around expanding frontal lobes. Diet is not a side variable. It is a structural driver. A Mediterranean network of early Homo populations consuming fish and cooking in earthen ovens creates a plausible pathway towards smaller teeth, lighter faces, and energetically supported brain growth. It also reframes the question of where our species truly began. A seafood-based cooked diet also intersects with brain energetics. The human brain consumes a disproportionate share of metabolic energy. Supporting that cost requires either high-calorie terrestrial hunting or dense marine intake. Coastal ecologies provide consistent yields. Fish are predictable, renewable, and nutritionally efficient. If Morocco and the Levant were connected through shared ecological strategies, then the gracile, cognitively sophisticated humans of later periods may have inherited a much older dietary template. Arginine-rich protein intake, enhanced nitric oxide signaling, and improved vascular regulation could have contributed to neural plasticity. While one must be cautious about attributing single amino acids to large evolutionary outcomes, diet unquestionably influences gene expression and developmental pathways. Over hundreds of millennia, selection operates on metabolic efficiency and enhanced growth of neurons in the frontal lobes. Cooking fish in earthen ovens 780,000 years ago is not just about food. It is about planning, patience, and metabolic redesign. It is about reducing the mechanical burden of chewing and reallocating energy toward neural tissue. It is about transforming ecology into physiology. In that transformation, one sees the deep roots of gracility, smaller dentition, and perhaps even the biochemical shifts that allowed our brains to operate at unprecedented levels. A population consistently consuming cooked fish would differ physiologically from one reliant solely on raw red meat and fibrous plants. Teeth would shrink, jaws would lighten, faces would retract, brains would expand within energy constraints made manageable by cooking. And so the scene by the lakeshore 780,000 years ago is not trivial. It is foundational. It suggests that the roots of our lineage were already experimenting with culinary technology deep in the Middle Pleistocene. It suggests that the Mediterranean was not merely a passageway but a cradle, a zone where early Homo populations blended African ancestry with Southwest Asian ecology. The Moroccan mandible from Thomas Quarry sits chronologically almost exactly alongside the Levantine cooking evidence. Two ends of a basin. Two populations perhaps connected through intermittent dispersal, both near the base of later human divergence. If these groups shared ancestry, then seafood consumption and controlled fire were not local accidents, but widespread behaviors. And if that is true, then Homo sapiens, in a broad evolutionary sense, may have been emerging not from a single geographic spark, but from a long Mediterranean arc of innovation. To cling to the idea that Africa and Eurasia were evolutionarily sealed off from one another is not just outdated. It reflects a failure to engage with basic geography and deep time ecology. The Afro-Eurasian landmass has always been connected, always dynamic, always permeable. The Levantine corridor is not a mythic gate, it is dry land. To imagine a pure, isolated African homeland from which humanity dramatically burst forth requires ignoring climate cycles, fossil distributions, and the obvious continuity of terrain. And bluntly, to think otherwise is intellectually careless. It rests less on geological or archaeological reality than on pure imagination. The persistence of a rigid out-of-Africa versus Eurasia dichotomy reflects a mental map in which Africa is framed as some untouched wilderness, a distant, unchanging cradle of wild beasts and primitive humanity, rather than as an integral, dynamic part of a single interconnected continent. That caricature, not the data, is what allows simplistic isolation models to survive. To imagine early humans evolving in a sealed African bubble and then bursting outward in a singular moment requires flattening the complexity of both geography and population structure. It turns evolutionary history into a migration myth with a clear beginning and a triumphant exit. But deep time rarely works that way. Thanks for watching.